Okay, well, good afternoon, everyone. I'm happy to introduce my PI, Dr. Eva Top. I'm going to give you a little background on her. She is a full professor here in the biology department at the University of Idaho. She's originally from Belgium, where she received both of her degrees at the University of Ghent. And her early work was in capturing plasmids in the wild. One of the things I have always admired about Eva is she's always pushing to improve her research by incorporating new models or new techniques or new technologies. And 20 some odd years she's been at the University of Idaho, that's really shown. She's worked on a lot of things, collaborating on modeling plasmid dynamics, looking at how biofilms or heterogeneous populations can influence plasmid dynamics, and embracing emerging technologies like DNA sequencing to conduct influential experimental evolutionary studies of plasmids. She's an ardent contributor to the peer review process as a reviewer, editor, and editorial board member of too many things to mention. She is the secretary of the International Society for Plasmid Biology since 2012, and she knows everyone there. She has served as the BCB director here at the University of Idaho, and she's a respected scholar as evidenced by the quantity and quality of her publications and her funding sources, which just in my time here has included a R01 renewal, several NSF beacons, and a sizable USDA funding. Beyond her scholarship, Eva is also a world-class mentor. From undergrads to postdocs, Eva treats everyone with dignity and respect. She's always involved and willing to make time and give support. So I consider myself very lucky as having won the lottery for the opportunity to earn a PhD in her lab. And now that I'm done laying it on real thick for my PI, can we all give a digital welcome to Dr. Eva Top?
Thank you, Clint. Don't make me cry, okay? So it's a real honor to give this IBEST seminar today. I understand that a few people were chosen either because they're upcoming stars or long-time IBEST faculty. And so I feel a little old today being among that second group. But just to show you how long I've been here, let's see if I can actually transfer to the next slide. There we go. So I've been here indeed almost 2 decades, and this is 2002 when we got the first NIH COBRE grant here at the university, which was basically the funding for the IBEST Institute, which was an initiative at the time. So you can all see we've all aged a little bit. I was trying to hide this. Okay, and so I'm really grateful to IBEST and to all that NIH COBRE funding, because without that I would not be where I am today. And I also realize I have collaborated with a lot of the people on these pictures here. I'm grateful for that spirit of collaboration and collegiality. So, okay, so just so you don't get lost in my talk, which could be a little dense, I tend to do that. I'm going to give you a little background first, and then basically an overview of our research questions, and I will briefly touch on the first main project that we have that's funded by the USDA, and then the second one I'll go in a little bit more depth, and actually not depth, but a broad overview of what we've learned so far. So just because not all of you are microbiologists, and certainly not plasmid biologists, I want to make sure we all understand that bacterial genomes are actually quite flexible, and it's not like they have one chromosome and they hold on to that and it never changes. Right? There are a lot of genes that are gained and lost over time in these very complex microbial communities, and bacteria interact with each other and exchange genetic information. And so the way I like to think of it is that some parts of the bacterial chromosome, usually there's one per cell, but sometimes more, are very conserved, right? And that is what you're familiar with. And they are conserved mostly because they're essential for the bacteria, and you don't want to mess around with those essential functions. But then on the other side of the screen here, you have a lot of traits that are encoded by genes that come and go, basically, and they are encoded on what we call mobile genetic elements. And I will focus on one of those types of elements. And these elements code for traits, as I said, that are not always essential, but if you don't have them, in some cases, you could really be doomed. Like if you don't have the right resistance to a certain antibiotic when you're in the gut of a patient who's taking that antibiotic, you may be doomed. So some of these genes are actually essential at certain times of the lifetime of a bacterium. And so I'm going to focus on antibiotic resistance as one of these traits, but as you can see, there's many more, and the list is much longer than that. And so of the mechanisms of horizontal gene transfer, there are three major ones, and we don't need to go into textbook details, but basically I'm going to focus on conjugation, which is a process that requires the cells to make contact, and a plasmid, which is one of these circular elements here that I'll get into, transfers from one bacterium to another. Basically a copy of it is transferred, so the donor pink cell here does not lose the plasmid, it gives a copy to the recipient. And so if that plasmid codes for several antibiotic resistance genes, that means that the yellow bacterium is now resistant to multiple antibiotics. As you can see, all these mechanisms have been discovered more than half a century ago, and we still have so many questions. So again, to bring you all up to speed a little bit, plasmids are extra chromosomal genetic elements that are usually circular, but there are also linear plasmids. They replicate separately from the chromosome. They come in either one copy or a huge amount of copies per cell. Most bacteria have plasmids in nature and in your own bodies. And so they are not cloning vectors, they are real natural plasmids. They come in a range of copy numbers, host ranges, meaning that some of them will only replicate in one type of bacteria, others can spread to different bacteria and replicate in there. And some of these, as I said, encode drug resistance, which we will focus on. Some of these are self-transmissible in the sense that they have everything it takes to transfer to another bacterial cell, which does not have to be the same species. And so since we found these plasmids both in the environment and the clinic, it is obvious today that they play a very important role in rapid evolution of bacteria, especially rapid adaptation to a new environment, such as a drug. And so just very briefly, right, the reason why plasmids have basically become popular again has a lot to do with the antibiotic resistance crisis and the fact that we're running out of options, that many of the antibiotics are not working anymore because bacteria have become resistant, right? And so there are these doom scenarios that we may enter a post-antibiotic era and all that. But basically, antibiotics have been extremely useful, and they are still very useful. But if bacteria gain more and more resistance, we could run out of options. So just a little history here, right? At a century ago, we still didn't have any antibiotics. I always, for me, it's easy to remember the First World War because I grew up in Belgium, which suffered a lot through that. There were no antibiotics, right? In the Second World War, there were antibiotics, and that made a huge difference. So I know, and since Bruce is here, he will make fun of me that I show these large numbers of deaths that have been predicted by this paper or other papers. They are doom scenarios, of course, right? We really don't know what's going to happen, but there's clearly a problem. And you may have heard of NDM1 and carbaphenemases. I will talk to those about those in the very end. Colistin is one of these last resort antibiotics for which we now have resistance genes on plasmids, and they are spreading. So the basic problem is, right, that's when you have an infection and a bacterium starts growing in your body. If it's sensitive, you can treat it with an antibiotic and the bacterium will die. But if it's resistant because it either has an additional gene or a few genes, or it has a mutation in its chromosome, it may not die and it keeps growing in your body and you need to go back to the doctor and get another antibiotic. So we are focusing not on mutations in chromosomes that cause resistance, but on the genes that are acquired by bacteria that add additional resistance, that confer additional resistance to the bacteria. Right, and so it's, again, not very new because already in the 80s, right, people were talking about the fear of these epidemic plasmids that they are spreading all over the world. But still, we were kind of confident, I guess, that antibiotics would do the job. Oops, what just happened? Okay. And so just a few years ago, another paper came out from our collaborator, Amy Mathers, again, using that term epidemic resistance plasmids, right? So we clearly have an issue of these plasmids spreading worldwide in these very successful clones of bacteria that are hard to deal with. But not only in the clinic do we find these resistance plasmids, they're also in the environment. And that's actually one of the major questions we have, is what is the link between the two, as you will see. And so soil, manure, wastewater treatments, the gut of any organism or any surface of body or cavity or whatever, humans, animals, plants, there's tons of bacteria and resistance plasmids often in those bacteria. So with that introduction, I just want to basically frame the long-term goal that we have in the lab is to gain insight in the ecology and evolution of these natural plasmids with a focus on resistance plasmids, and not just in pure culture, but also in their natural communities. And the reason that we want to understand that better is, one of the reasons is of course also just purely out of scientific interest, but also because if we understand these dynamics better, then we may be able to kind of slow down that spread of resistance plasmids eventually and avoid them from entering the hospital in these nasty pathogens. And again, this is not an idea that I came up with necessarily. There are other groups that are thinking of focusing thinking about the ecology and evolution of bacteria and not just trying to kill them, but maybe outsmart them, which may be impossible, but it's worth a try. So I have two major questions, and I'm going to touch on each of those. So first of them is really thinking about this link between environment and the clinic, is what are the reservoirs of antibiotic resistant genes out there, and also the plasmids, and how do they transfer to pathogen, when, where, what are the factors that promote that transfer? So that is one main question. And the other one is, once these plasmids do end up in a new bacterium, either a commensal in your body or a pathogen, what does it take for these plasmids to really be maintained so that the resistance genes on these plasmids are retained, which is not what we want, right? But we know that bacteria are very quick at adapting and plasmids are quick at adapting. And so what are the mechanisms that stabilize these plasmid host relationships? Because maybe we could use those as a target to intervene. And then within that third one in gray, because I don't have time to talk about it today, is the effects of spatial structure. For example, in bacteria growing in biofilms where they are not completely mixed. What is that effect on the spread of plasmids and on the evolution of plasmids? That's a whole other discussion that we don't have time for today. So let's start with the first one. So basically, when agricultural plots are fertilized, there's of course chemical fertilizer, but there are also two forms of organic fertilizer used in agriculture. And one is biosolids, and it's probably the one that some of you may not have heard of. So it's basically the waste product of wastewater treatment plants, which is all the microbes that degrade your waste when you flush your toilet. They are basically biomass, right? That is growing and growing, and so one needs to take care of that. So it gets concentrated, dewatered, and then composted, and it's nowadays used as a fertilizer in agriculture. And the other one is, of course, manure, that you're much more familiar with, not just from cows, but from any animal that is grown in huge amounts these days. And I just wanted to point out that Idaho is actually the third or fourth largest dairy state in the country right now. It competes with New York State for the third place, apparently. And I found in an article recently this year that more than a third of Idaho's agricultural cash comes from the dairy industry. So it's really booming. And so there's lots of cows that produce lots of manure. And so what do we do with that? We put it on land. And so first, to go back briefly, as I said, I've tried to give kind of an overview of what we've been doing in these years. I can't cover everything, obviously, but the biosolids that come from the wastewater treatment sludge. So what we showed is that what we wanted to know is, do these biosolids still contain live bacteria that can still transfer their resistance plasmids to either commensals from your body or to pathogens? We know there's plasmids in there, but are the bacteria still alive enough and active enough, right, to donate these plasmids? And the answer is yes. And so when you mix these biosolids with some bacterial strains, including a pathogen like Salmonella typhimurium, the Salmonella will pick up plasmids from that biosolid material. And so this is a paper that's still pending any day now, I hope. Should be accepted. But so the bottom line is that we did find plasmids in there. I'm not going to go into this, but the yellow genes here are all resistance genes or transposase genes, meaning that these plasmids have up to 5 different antibiotic resistance markers, 4 different classes in some cases of antibiotics. So, you know, you can imagine when a pathogen picks that up, that they're much better off than a minute before. And then the manure question, right? So manure is even used much more as fertilizer. We know already from the literature that there are resistance plasmids in there. So our question is really, do these plasmids then eventually end up in the soil or in the groundwater or in the river water, creek water, because they transfer to other bacteria? And so here I am borrowing a slide from Chava, who gave this wonderful presentation earlier this week, right? So animals, the reason why there's resistance plasmids in the manure of these animals is because for many decades now, we've been feeding them low amounts of antibiotics every day because that promotes their growth and avoids infections. And so when you then use that manure, so I'm going to skip that first part on the top there, but when you use the manure of these feedlot animals on soil, the question is, do they transfer to soil bacteria, to water bacteria? These plasmids, do they transfer, right? Because if that is the case, then those bacteria could eventually come in contact with us and transfer the plasmids further to pathogens. So this is the big question, right? We really don't know if this is a dead end when you dump manure on a soil, that the plasmids in those bacteria just disappear, or do they spread and form like a new reservoir in the soil? And so, as I said, right, this is basically the question of Chava's thesis, who gave this excellent prelim presentation today and did a great exam too, right? And so what he's focusing on is using this novel method, at least novel in the sense of its application here, to see if we can monitor the spread of a plasmid in a real very complex bacterial community like a soil. Because that has not been possible so far. And so, again, I'm not going to go into detail because he talked about that on Monday. Some of you were there. And also, if you were not there, then Thibault will give a seminar in biology. So here's a little plug for him on January 22nd. And you'll probably hear more about this famous high-C method. But I'm going to just mention the highlights of it. So the problem today with trying to monitor where resistance plasmids go from manure or from these biosolids is that we are either limited by growing the bacteria, cultivation, and that is not very good because we know that about 99% of soil bacteria cannot be cultured in the lab. So that's a huge limitation. You could say, well, just sequence all the DNA and you'll figure it out. Now that's not true because the metagenomics approach, where you extract all the DNA and then blast it and sequence it, that loses completely the link between a plasmid and its chromosome of its host, right? So you don't know, you may be able to reconstruct plasmid genomes and reconstruct chromosome genomes, but you don't know who belongs to whom. And then there's some other tricks, like scientists who have marked a plasmid with a fluorescence protein gene and then monitor it that way. But that is, of course, artificial because you are modifying the plasmid. Not every cell in the soil will be able to express that fluorescence. So there's issues there too. So the nice thing about this high-C method is that it really, in the whole process, maintains the link between the plasmid and the chromosome within a cell. And so we are hoping that this will help us understand better who are the bacteria that contain certain plasmids, and are they receiving it from manure? Are they then retaining it later in the year, right, when they become regular soil bacteria? Do they hold on to that plasmid? And so I'm not going into the methods itself, but basically when you have like an orange bacterium with a green plasmid and a blue bacterium with a gray plasmid, what I'm saying is that with regular metagenomic sequencing, you cannot reconstruct that pair, while with high-C, because you basically freeze the DNA in place, you cross-link the DNA in the cell before you lyse the cells. And so now you can reconstruct, when you sequence little fragments in the end, that this plasmid is connected to this chromosome, because we have one read that has on each side a little bit of that plasmid and a little bit of that chromosome. So if that doesn't make sense, then go listen to Thibaut's talk in January. But the bottom line is that we are expecting that this will be a way to really like follow the trajectories of plasmids throughout rather complex microbial communities. So that's all I'm going to say about that question, but it's clearly in its infancy, and so we're hoping to make great progress on that. But the second big question here, keep track of time, okay, is, okay, so these plasmids are spreading. Sometimes when they get into a bacterial host, they're very well maintained, even in the absence of antibiotics. So they don't need any selection pressure, apparently. And in other bacteria, they're lost very rapidly. And so what is the difference, and how does a plasmid adapt to a bacterium? How does a bacterium adapt to a plasmid? As I said, if we understand that better, maybe we could use these mechanisms as targets in future therapies that are not just killing the bacteria. So before I go into how we address that question, I want to explain to you what we mean with plasmid persistence. So bacteria grow in giant populations of often millions and billions of cells. We're not talking about individuals here. But when a cell divides, then the question is, will these plasmid copies, like 3 here in this example, end up in both daughter cells? Or will, by some chance, one of the daughter cells get all the plasmids and the other not? If that's the case, you have loss of plasmids, right? And I'm using immediately some Greek letters here, and this is for Bruce, who is here, right? So the beauty is also that you can model these scenarios, because it can be simplified to a few mechanisms. So even now we have these 2 bacteria, one with and without plasmids. What happens, and what's often forgotten, is that the plasmid-free cell usually grows a little bit faster in the absence of antibiotics, because it doesn't have to maintain these plasmids, it's not expressing any proteins on these plasmids. So in this example, you have like 3 generations, while there's only 2 in the one with the plasmid, right? So it gets an advantage and it can outcompete plasmid-free cells. But don't worry, because there's also conjugative transfer, and that means that the plasmid-containing cells that are still around can reinfect the plasmid-free cells. And so in the end, you have this balance between loss of the plasmids, the cost to the fitness of the host, so basically the growth rate, if you want to simplify it, which is not the same, similar. And then you have the transfer rate, which can resupply the plasmids to plasmid-free cells. And so this whole process here we call plasmid persistence. And as you can see, there's these underlying causes for it that are independent mostly, and can play a very important role. So how do we measure persistence? Well, very simple, but very primitive still. We just grow bacteria in the lab in test tubes in the absence of antibiotics. And then we plate them and we pick colonies usually, and we figure out which ones are resistant and which ones are not. And then we confirm if the resistant ones indeed still have the plasmid. That means they are maintaining it, and the ones that are sensitive have lost the plasmid. Right? And so what we see is, in the case of low plasmid persistence, when you plot this kind of persistence assay, you have a time in the x-axis and a % of P+, which is plasmid containing cells, in the y-axis. And so an unstable plasmid will show like the yellow profile, and a very stable plasmid with a high plasmid persistence will show the green profile. So that means that all the bacteria keep the plasmids, sometimes for a very long time, even though there is no apparent positive selection for these plasmids. And so what we do is we look for bacteria plasmid pairs that are showing this low plasmid persistence profile, and then ask, do they rapidly improve when you allow them to stay together for a while? So how do you study bacteria, the coevolution between plasmid and bacteria? We were inspired in that COBRE grant in the year 2001 by studies from the Lenski lab, and actually Rich Lenski was I think a student or postdoc of Bruce Levin, right? So we have actually the real ancestor here of this whole of these studies. And so we were inspired by these studies that showed that there was some adaptation between plasmids and bacteria. Molecular mechanisms were not yet identified at the time, right? Sequencing was not really plausible at these large scales. Since 2000, since we started doing this work, then a lot of other groups followed, and there's actually a ton of literature now on this topic. But still, very few molecular mechanisms have been identified. It's like, how do bacteria really better retain a plasmid over time? It's still very unclear. So what we do to try to get to this question is we evolve bacteria in the lab. So again, a rather simple system with test tubes and bacterial cultures. So now we grow them for a really long time in the presence of an antibiotic so that there is pressure for the plasmid to be maintained. We do this for a variety of times, sometimes only 30 days or 300 generations, sometimes 100 days or 1,000 generations. And then at the end, we ask, okay, are there changes in these evolved bacteria and plasmids? Where and what do they do to the persistence of the plasmid? And so the first question is indeed, right, has the persistence improved? Right, so we have this unstable plasmid at first. Over time, does it become more stable? And if so, why? So then we ask ourselves, is it because the plasmid changed or because the chromosome changed? And the nice thing with bacteria is, of course, you can freeze them, right? You can go back in time. You can also take the plasmid out and move it in the ancestral host, or do the opposite, take the ancestral plasmid, move it in these likely evolved bacteria, kick out the other plasmid. And so then you ask again, okay, when I have combinations of an evolved plasmid with an ancestral host and vice versa, what do I see? Right? Can I explain all the improvement in persistence by changes in the plasmid or changes in the chromosome? And then, or sometimes in reverse order nowadays, because sequencing has become so easy, is we basically sequence the whole genomes now, right? It started when we wrote a proposal of sequencing one gene, and then we went to, oh, let's do the whole plasmid. And now it's like, no, don't even take the plasmid out, just sequence everything, which is also by itself a very interesting evolution. So what are the genes that have changed? What do they code for, right? The function. Can we make sense of how that would improve plasmid persistence? In some cases, we reconstruct the mutations, so we are really sure that that's cause and effect. And then the next step is really getting back to that overview of all these underlying mechanisms of persistence. So if we do see improvement of persistence, is it because the plasmid has changed its cost to the host fitness? Or is it because it's not lost as rapidly anymore when cells divide, right? So that segregation mechanism is better. Or is there an increased transfer rate so that even when the plasmid is lost, the host can get it back through horizontal gene transfer? So we've done a lot of model fitting of these persistence curves, trying to figure out what's the cause here. And I'll get back to that in a minute. And then finally, all of this is interesting to plasmid biologists, but more generally, if you find a mutation that explains the persistence in one bacterial host of one particular plasmid, that's all good. But can you generalize that, right? Because is that same mutation going to help another bacterium plasmid combination? Yes or no? So what we then also sometimes do is, okay, if we had a mutation in a plasmid, like here on the left, when we move the plasmid in other bacterium, that can be a totally different genus even, is that plasmid also stable in other bacterium more than before it had that mutation, right? So that would be a change in host range of the plasmid. Or if we find mutations in the chromosome of the bacterial host, when we put another plasmid in that host, either with or without the first one, do we also see that that plasmid is now more stable? So is that bacterium becoming a more permissive host for all kinds of plasmids, which would be problematic in the case of resistance plasmids, when one mutation in the chromosome of a bacterium could allow it to hold on to a whole host of resistance plasmids. So just one plug for, again, how IBEST has promoted interdisciplinary work, and how I really have the best memories of collaborating with the mathematicians, and of course still do with some of them. So the first paper, I guess, was 2004 here in the good days when Paul Joyce was the PI of, or the major professor of Jose Ponciano and Zaid Abdo, who were the first BCB students. I think Zaid was actually the first BCB graduate. And so we started working together, and I just knew enough math not to be lost, and they knew enough biology to understand what we were trying to do. And so with some very basic start of, what is it that we're trying to describe here, the loss, the cost of a plasmid, and the transfer of plasmid between bacteria, we actually made a lot of progress in a very short time. And now I'm still working with Jose Ponciano on our NIH grant to further try to get a grip on these complex bacterial plasmid dynamics. So again, keep an eye on the time here. So I was trying to summarize all the things, all these evolution experiments we've done and what have we learned, right? And I realized that we've done a lot of them, and also especially we've had a lot of people involved, as I will show you in a minute. But so the whole idea is, if you have to do this with a lot of bacterial plasmids to try to figure out, is there any pattern, right? Do we always see the same changes that stabilize a plasmid in the bacterium, or is it all over the map? What lessons can we learn, right? So we use these different bacteria here. A lot of them came from the environment at first, and now more recently we are also using true opportunistic pathogens like Serratia marquescens from the hospital environment. And so we used always the same group of plasmids, and this is also a little plug for another IBEST collaboration with Jack Sullivan at the time, trying to improve our understanding of the phylogeny of this group of plasmids. So they have a very broad host range, so they are found in a lot of different bacteria. And so we just focused always on that group of plasmids. That's just our pet plasmid group, I guess. So here are just, and I probably forgot some, right? But all the people who have been transferring cultures for many days, and you have to come in always around the same day, weekend or no weekend, right? And so very grateful for all those people, and a lot of undergrads that I don't have pictures of or wouldn't fit on this screen have also contributed, more than the few that are on this page. So just very briefly, this group of plasmids has a very broad host range, so we call them also promiscuous plasmids. They transfer and replicate in different classes of bacteria, so that is already quite a reach. They will even transfer to eukaryotic cells in some cases. They just can't replicate there. The first plasmid that was described was actually from a burn wound, a Pseudomonas aeruginosa isolate. So they are found in the clinic, not very often, but it's not to be excluded. They are very common in wastewater treatment plants and soil, and occasionally also in manure. So what did we find, right? So in trying to summarize, the first question, when you expose plasmids and bacteria plasmid pairs to antibiotics, then you remove the antibiotic and you ask, is that pair more stable now? The answer is always yes, right? So we always went from an unstable plasmid host pair, where the plasmid would get lost over time, to a much more stable pair. So that's good. And this is just also to remind myself to not forget the honorary members of the top lab. We had Hannah George here. She's from UW, from the Ben Kerr lab. She graduated last year. It's just one of the thousands of examples we have by now, right? That your plasmid is very unstable. These are the real data. And after, when you test them after they have evolved over multiple generations, sometimes not so many, you see a variety of patterns, but clearly improved persistence of these plasmids in the absence of any antibiotics. This was a Klebsiella bacterium, so Klebsiella pneumoniae, which is problematic as a hospital-acquired infection. Okay, so how do they do that all? And what are the mutations that we see? Okay, this is a way too busy slide, and I'm going to cover it up for you here. Okay, so I'm not going to go through every detail, but basically the bottom line is there's no clear pattern, unfortunately, maybe, but also not surprising given the complexity of biology. So in some bacteria, we see mutations in the chromosome only that explain all the improvement of plasmid persistence. In some bacteria, we see mutations in the plasmid that explain all the improvement. And in some bacteria, we see both. right? So there is no one answer. So P or C stands for plasmid or chromosome. And then the question is, okay, where are these mutations, right? And are we sure that they are related, they are causing that change in persistence of our resistance plasmids? And here again, we have a whole series of options, right? We have chromosomal helicase-like genes that are mutated. We have loss of complete big sections of the plasmid that are responsible for the transfer, the horizontal transfer, or just a mutation in one of these genes. Or we see other changes, SNPs or indels in replication gene of the plasmid. Or we see whole transposons being acquired and jumping around, and so on. And so if I have time, I'm going to try to get to these last two examples, which are the current ones. But the rest, what I want to say still is that when we try to explain then how, right, what is the underlying cause of this improvement in retention of the plasmid, it's often the fitness cost of the plasmid that is decreased, but not always, as you can see. And in some cases, we've gone further and looked, okay, like in that first case, you have a chromosomal mutation. When you bring other plasmids in that bacterium, is it also retaining those better? And sometimes the answer is yes. Or when you have mutations in the plasmid, does that affect its host range? And the answer is yes. We've seen expansions of host range where more bacteria will now retain this plasmid better, or sometimes it's specialized to certain hosts and we got more like a host range contraction. So all over the map, as I said. So what can we conclude from that? Clearly, it doesn't take long, and we actually have seen now sometimes just 50 generations, which is nothing for bacteria. You can have a rapid adaptation of plasmid to the host or vice versa that improves the persistence of these resistance plasmids. But there is no clear mechanism, right? It's totally plasmid and host dependent. There's also often the fitness cost amelioration, so meaning that the plasmid is less costly to the host, which makes sense, that it will then retain it better. Also, some of the chromosomal changes we see, there may be some pattern, maybe because several of them also from other studies now point towards regulators, sometimes global regulators that are involved in the maintenance of this plasmids indirectly, probably, right? So that is an interesting avenue. And then also I want to point out, just more in general, right? All these studies are done in pure culture, often in nicely shaken liquids, very artificial. And so when you see loss of transfer genes, I ask myself, that's probably an artifact of growing a bacterium all by itself. Because if these bacteria are in communities of hundreds of other species or thousands, like in soil, I would think that the ability to transfer to other bacteria is probably what saves them, right? What allows them to survive when their host dies, and they escape just in time, they will live. So this was a summary of studies that were all done in the presence of antibiotic selection, right? So you're trying to maintain the combination of host and plasmid, and then you ask, okay, what has changed? But now what if you just grow bacteria with their plasmid and don't apply any selection? Many of you will say, well, plasmid will get lost. Well, true in some cases, but not always. And so I have a few more minutes, so I think I can do this. So Clint here, who gave this nice introduction, has been beating his head against the wall a little bit for this interesting phenomenon that we found. You grow E. coli with a plasmid from a Bordetella pertussis, actually from a whooping cough patient who died from it, unfortunately. You put it in E. coli, and you see the plasmid persistence is not great, right? So the plasmid fraction was high at day 0, but it decreases over time. What he did is take some colonies from day 5 after plating, and you re-inoculate those. So you grow them up with antibiotics again to try to start close to 100% there. And so what he shows now, when you grow them in the absence of antibiotics, they are almost perfectly persistent. And so these 3 different clones from independently evolved populations here have similar behavior. And so the question is what happened, right? And so what we showed is that it's clearly changes in the plasmids, not in the chromosome. And I skip over this, right, as if it's nothing, but you sequence the whole bacterium and you see literally 0 changes in the chromosome. and you do see a change in the plasmid. So what did they see is that in this plasmid, there were deletions, and guess what, of course, in genes that have an unknown function, right? So there you go, right? There's something very important to the persistence of this plasmid in an E. coli bacterium, but yet we don't know what this gene is doing. So in some of these clones, there was 2 genes deleted and some just 1. So we are focusing on this 1 gene now that is clearly the culprit apparently, and really makes the plasmid very non-persistent in E. coli. Based on the sequence, it's a putative methylase, so that's what Clint is beating his head against the wall for, is trying to figure out if that's really true, right? Sequencing is easy, but then confirming what is it really doing is another story. But this is very rapid, right? So 5 days of growing in the lab, and under other conditions, you could even get there in 2 days probably. You see this plasmid change from getting lost to getting lost much, much more slowly. So another last story then before we stop is something that Erin is working on, Erin Mack. So we started to collaborate with Dr. Amy Matters at the University of Virginia Medical School. And she has all these hospital isolates that have tons of plasmids. And she focused on KPC, which is a carbapenemase, right? It's a beta-lactamase. So it's the enzyme that cleaves beta-lactam antibiotics. And carbapenems are these last resort antibiotics. And so there's great concern that when bacteria are resistant to those, then what do we do? So this is why we avoided working with clinical isolates for a long time. This thing has 5 plasmids in addition to a chromosome. Right? And so the KPC gene, which is that gene of interest here, is on one of these plasmids, 70 kb plasmid, so it's pretty large. It has 2 origins of replication. It's also a complication, right? So we don't even know which one is active and which one is not, and maybe they're both. Serratia is one of these nosocomial infections, right? So it's a hospital-acquired opportunistic pathogen that in the last decade has come up as an increasing problem. And so we were asked, we wanted to understand, okay, when we grow this bacterium with or without the carbapenem, what happens to this suite of plasmids? And the reason why we were interested in that is because Amy and previous studies had shown that this KPC gene, so the beta-lactamase gene, is found on transposons, and just like in eukaryotes, transposons can hop from one place in the genome to another, and from plasmid to chromosome, and vice versa. And that they found the same gene on the same transposon, but in different color plasmids here, so in different plasmids, and that there were transposons within transposons, and these were found in isolates from multiple patients in different places. Right? So this thing is going around and is a problem. So we grew this Serratia strain in the presence and in the absence of antibiotics, and we are in the middle of this study. So I just wanted to give you a little primer because I thought the result is counterintuitive and kind of intriguing, I think. So you have this bacterium. Here is the KPC gene in pink, right? So if we grow this in the absence of meropenem, the carbapenem, what we get is we lose the plasmid slowly, but if you look here where my cursor is day 10. So most assays we do are 10 days or less, or 7. We would have thought that's pretty stable plasmid, right? But then if you go longer, it does drop out, but still, you know, if you have 1 billion cells, you still have more than 1,000 left here in your population, right? So it's not gone, the plasmid, or the resistance, I should say, it's not gone. So this is the fraction of resistant cells. So when we do the same, of course, in the presence of meropenem, no surprise, right? The bacterium has to hold on to its resistance gene, so the resistant cells remain at 100%. And I just drew the line there, even we don't need to show the data. It was just 100% all the time. So then we ask, okay, what happened to the plasmid pattern? And so in those rare, rather rare, but still numerous clones that were grown for a whole month without the antibiotic, we sequence them completely and we see no change to the plasmid. profile, exactly the same. Also, when you do this, you extract the plasmid DNA and run it on an agarose gel, exactly the same pattern as before. However, when you take some of the clones from the meropenem grown cultures that was under strong selection with a high meropenem concentration, something changed. What we call the KPC plasmid that originally had that KPC gene is gone. So I drew it on the side here. It's missing, but the KPC gene is still there. And so again, with sequencing and puzzling, we think that what happened is that the gene hopped onto one of these small plasmids, the 6 kb plasmid here. And so now is a 16 kb plasmid because that transposon is about 10 kilobases large. So that has consequences, right? Because you think, okay, we're not having meropenem, let's say in a patient in a hospital, wherever, for a whole month, but yet all you need to do is apply antibiotics again, and this bacterium comes back with all its plasmids. While if you had applied strong meropenem selection, you would still have the gene, so it's still a problem, the bacterium is still resistant, but the nicely mobile plasmid that it was on is actually missing now. So we are in the middle of trying to figure out how mobile is this resistance still after it's jumped on this smaller plasmid. So we are also doing Oxford nanopore sequencing to really confirm this, because this is a little bit preliminary still. So this is basically all I wanted to say, and I'll skip the details of that. And so I have a few take-home messages, I guess, after all that work that we've done, is since we're in the middle of it, it's more than time to try to understand if plasmids really do travel from farm to clinic, right? And how and where can we slow them down? We know that plasmids and bacteria rapidly evolve and rapidly improve plasmid persistence, even when there is no antibiotics present. And so what that means is that resistance genes are now retained better, which also means they can further spread more likely to other bacteria. This last thing I showed you, unexpected effect, right, of high antibiotic concentrations. So the gene survives, but not the plasmid. So how should we think about that? I would love to hear some feedback on that phenomenon. And then that some of these chromosomal mutations really do lead to improved plasmid permissiveness, like the bacterium is now able to retain other resistance plasmids much better than before. And so that can create a reservoir of multiple resistance genes. But so far, we don't really see one mechanism, right? One molecular mechanism or one even an evolutionary mechanism by which this all happens. So we're still not really much closer to knowing how should we target these bacteria in order to make them stop transferring plasmids or retaining plasmids. And also, again, that I really appreciated the help and the collaboration with the mathematicians to get a better grip on the underlying mechanisms behind these dynamics, and also to determine statistical differences between. So that is basically all I wanted to say. And there's way too many people to thank, of course, right? So I went back my CV actually to, and I may still have missed some lab members. I cannot list all the undergrads, there's more than 50. I also found some old picture which made me reminisce of the first lab group we had. Some of you remember Stacy and Lean, I'm sure, and maybe Masa Sota. This is the last picture we have. We urgently need a new lab group picture. It's 2 years old. We've been really fortunate to have lots of funding and lots of support from IBEST and all the wonderful people in the genomics core. And also I have a lot of collaborators, right? We cannot do this on our own anymore. And so I really appreciate all of them. And then, of course, the people who do all the work, right? The current top lab, it's a fun group, as you can tell. And yeah, we couldn't do it without them, of course. So great. Many big thanks to everyone. And thank you all for staying on here for this long. Questions? I'm not very familiar with this. Let's see if I can see you somehow. There we go.
That was a great picture of Jack.
Of who?
Jack Milstein.
Oh, you mean the beginning?
No, that last one with the baby.
Oh, Jack Milstein. Yeah. Yes, that was a great picture. Yes.
Hey, Ava,
Great to know that there's so many delicious questions that are unanswered about plasmids. And I recommend Stan Falco's memoir, because he did, I don't know, the early work. When I read his book, I just, the whole early period showing that so many of the resistance is coded for by plasmids, and totally changed everything. And I recommend that.
Good.
Ava, there's quite a few questions.
Oh, I need to look in the chat. Yeah. Okay. All right.
If you want to just read, or I can read them out loud.
No, that's good. How many different plasmids do bacteria typically have? So this is also all over the map. As I showed you in the end there, right? That Serratia strain had 5. Sometimes it's just 1. It can be more than that. There is really no, I don't know what the median is. We don't know, of course, because we haven't looked at all the bacteria and all their plasmids. But it's not uncommon to have more than 2 plasmids or more than 1 plasmid in a bacterium. How fast do plasmids carrying antibiotic resistant genes spread? How does it be based on the plasmid in question or species of bacterial host? Yes, good question. So how fast is, of course, a very difficult question to answer, because it depends on where, on the environment. Like if you test this in the lab, the plasmids we work with, you come back the next morning and you have 100% plasmid transfer, meaning every recipient of the billions of cells will have the plasmid. So there's a very high transfer rate. But you put these bacteria in the soil and it will be a lot slower. And so there is no one rate I can give you. It does depend a lot on plasmids, on the plasmids, obviously, but also on the bacteria and even the bacteria combination. So some donors will be great towards one certain recipient, but not to another species of bacteria, or vice versa. Some bacteria will receive plasmids from one and not from another. Chris marks, to answer the number of plasmids question, there can be, this is an answer maybe, okay. Yes, so, and also the number of plasmids, yeah, you have to be careful. So I was talking about the number of different plasmids in that first question. If you're talking about the number of copies for one plasmid, that is also very variable, which is what Chris was answering here, I think. From the sequence, do you know that the KPC has moved to unique plasmid or recombined out a portion with the gene and deleted the rest to reduce fitness cost? Okay, so you are Aaron's PhD advisor, right, David? Yeah, so she's actually in the middle of that. Unfortunately, she's not here because her partner has his birthday and she had promised something to him a long time ago. So.
I was just wondering whether you'd had a chance to sequence that yet.
Yes, so we did sequence.
Especially with two origins in there, it seems like it could have easily recombined.
Yes, so we lost both origins, confirmed by PCR and by sequencing. So the plasmid is really gone, at least that part of the plasmid. And the KPC gene, when she aligns it, the problem is that transposon, because it's so big, right, it's jumped into another transposon that is on more than one of these plasmids. So in some cases, we can't say for sure yet, but in one case, it jumped in a gene of that small plasmid. And so that's why we need to do the Oxford nanopore, the long read sequencing, to really be sure where did it go. But it looks like that large plasmid is mostly gone.
Okay, yeah, thank you.
Then am I understanding the last part correctly? Multiple plasmids within a bacterium can also transfer genes between plasmids. Yes. Does that happen often? Right. Often is always hard to answer, right? So yes, it happens. How often? How important? I think quite important, because as I said, you find the same resistance gene on different plasmids on often exactly the same transposon. So they do hop between plasmids. That is also the way for transposon to get out of the cell into other cells, because transposons on their own cannot get out of a bacterium, but once they're on a plasmid, they can get out. What do you think about the more recent solutions to tackle the antibiotic resistance problem, like the use of bacteriophages and other technologies? great question. We have Bruce here. He knows a lot more about bacteriophages than I do. I know, I know. Of course, I think we need to look at everything, right? So this is like climate change, right? You have to not just say, oh, we can't do windmills. No, we have to do windmills and consume less energy and do this and do that, right? So I think it's the same with antibiotics. On multiple fronts, we need to be prepared, I think, because there could be a real problem in the future. I think phage have shown their usefulness in some cases. What was the other one? Other technologies. Yeah, I don't know too many other technologies. I know CRISPR, talking about CRISPR, right? So some scientists have shown that you can actually use CRISPR to get rid of bacteria that have a very specific gene or plasmid because you target that sequence. It's a possible future technology. I think we should look at all of these. Thank you, Dr. Top. Very nice. I'm curious that have you guys found other sequences from other organisms in the Hi-C method? Do bacteria also integrate genes from other organisms? Thank you. Okay, so the nice thing about Hi-C is that it sequences everything, right? So we have not, so Thibaut Stalder has shown that when you take a wastewater sample, you can link up certain plasmids with certain bacteria in that wastewater sample. So other organisms, I guess you mean other than the ones we were focusing on. So this is still early in the project, right? So we are going to follow the plasmid and see if it goes to other bacteria. We haven't done that part yet. We're optimizing the method, improving the detection limit right now. But definitely, yes, bacteria integrate genes. So that's a good point. Plasmids don't have to stay as independent little circular molecules. They can integrate in the chromosome, or the transposon can hop into the chromosome. Part of the plasmid can integrate. There are also integrative conjugative elements that do just that. They come out and go in the chromosome, and then they can conjugate and transfer to another bacterium and then integrate. So the possibilities are endless, I would say. Any other questions? It is 29 after.
I haven't really kept up with plasmids as much as I should, but the average E. coli has about 3 plasmids, and maybe a third of them are big enough to be conjugative. Nevertheless, it's not clear what these plasmids are doing. I don't know, does anybody, you know, you have the multiple resistance plasmids, which we all like to study, but what are all the other plasmids doing, and why, why are they clustered there?
Right, it's the same with that serratia, right? What are these other plasmids doing there? I mean, it's not obvious. And I think Amy Mathers has actually shown some interesting things, that these little plasmids just go along with the big one. So when you're selecting on transfer of resistance, you move the big plasmid and the little ones just go along. And so why? And why don't they all become one big plasmid, you could ask. Right? There's lots of interesting questions there. It provides flexibility, but then, like you say, you do see multiple plasmids. So is that just a moment in time when these plasmids are somehow useful, or are these plasmids really so addictive that they don't get lost? Chris, did you have a question?
I did have a question, though. That makes me also think. It seems when looking at very closely related strains of the same organism, they very rarely have the same set of plasmids. I mean, sometimes like the symbiosis plasmids of rhizobia or something like that might be relatively well conserved, but often nearly identical strains genome-wise can have totally different plasmids. So it suggests, you know, quite a lot of flip-floppy. And so whether they necessarily are doing anything, or they're there because they can be there, just like what is a virus doing for a cell? Sometimes the answer is something, but often it could be nothing at all.
Oh, exactly. Yeah, I like to think of plasmids as kind of genetic parasites, right, that occasionally provide an advantage, just like a phage, probably.
Yeah. So the question I had, and I know that there's some interest in that you guys have had, Clint in particular, in this direction, but I couldn't remember kind of what the hook was for it. Is there any evidence for a role of epigenetics in plasmid maintenance, in particular, say something like DNA methylation, where you could get an epigenetic selection for a different epigenetic state that would change plasmid, any one of those traits, maintenance, exchange, etc.
Yeah, it's a good question. Yeah, so far we always just resequence the bacteria, and if we don't see a chromosomal change, we assume that's not explaining what we saw, right? And it's usually true because the plasmid change would explain everything. But we haven't looked into that, and the reason why Clint is interested is because that gene that got deleted codes for potentially a methylate.
That's right. That's right? I knew there was a reason. I couldn't remember what it was. Yeah,
right.
Very cool.
Confirm, is it really
restriction modifications often coded for by plasmids.
Yes. Yes.
And so the plasmids are playing that kind of protective role. I mean, they're,
Yes.
I mean,
yeah, but this is
defending their turf.
It's a long story. Yes, this methylase is not one of those restriction methylation pairs, right? It's a dam methylation. But yeah, there's like these INCP plasmids that we work with. The reason why they're so good in so many hosts is they don't have a whole lot of restriction sites actually for the common bacterial restriction enzymes, so they don't get chewed up when they enter.
Selected out. Yeah.
Yeah.
Very cool.